This is Mary from Open Helix with this week's Tip of the Week. If you're seeing this tip someplace else, be sure to come back by our blog at blog.openhelix.com, where I'll have additional details. I'll have links to all the things that I'm talking about today and guidance for you to proceed the same way that I'm going to be showing you in this quick tip. So check us out at blog.openhelix.com. So for today's tip, I am going to be talking about how you can access the ENCODE data. Now, as you might have known in the last week or so, there's been a media blitzkrieg, as I mentioned here on my blog post, and a backlash by scientists about some of this the claims that are made by the ENCODE team. But I hope that this doesn't dissuade people from actually getting in and using the data, because I really think that ENCODE can enable smaller science. If you have a topic that you're interested in, you may find new data in these genomic regions that the ENCODE team has generated. I want you to be able to check this out, and that's what I'm hoping to illustrate here today. Come by our blog post, read this, and I'll give you more details about how to do that. But what I wanted to illustrate first is that one of the things that can help you is that we've got a tutorial that explains step by step how you can take your browser and start looking for ENCODE data in the UCSC Genome browser. And so this tutorial is available for you. All you have to do is click the launch button here and you'll get a movie, and it will take you in a series of chapters here through ways to access and show this data on a browser. So you can look around at your favorite genomic region and see what's going on with ENCODE data added. You can come by and take a look at that tutorial. I would recommend that. Then after that, what you can do is turn to the UCSC Genome browser. So here's a UCSC Genome browser that I've got with a gene region here. This is TP53, and I show you exactly how to get to TP53 and the exercises that go with that tutorial. So you can go to this region, and then you can zoom out a little bit and look at, there's a nearby gene here, there's another gene right near there. You can start looking at the data that's available for you in the UCSC Genome browser. By default, when you first arrive now, there's ENCODE data. So here you can see there's ENCODE data here. There's a couple of other kinds of data tracks here that are available for you that are ENCODE data and show you that. Now, there may be more things that you want to do. There may be other types of data that you want to illustrate, or you may want to look in some of these in more detail. For example, maybe you want to look at the transcription factors that are in this region. I show you in the tutorial how to go to that track, and that track would be here in the ENCODE regulation data set. I can show you how to turn this on in full right here, click submit, and you can go back to the browser and see in the new view here, you can see all of these different transcription factors that were all compressed into one dense line before, but now you can see the different transcription factors that have been shown by the ENCODE team to be associated with this area. Now, of course, you want to turn on the controls. You want to look around at other cell lines and other ways to look at this data. But this is how you can get started looking at the ENCODE data in the UCSC Genome browser. And then maybe you found some data that you thought was interesting, and you think that in your genomic regions of interest, you might want to know more about whether this is valid or not. So another thing that you can do is you can go over to the Nature portal that provides you access to all of the data and papers from the ENCODE project here in this ENCODE Explorer. So what you can do here is you can take a look at the different types of data that are available. And maybe if you are interested in transcription factors like I showed, you would want to go to this transcription factor motifs thread. So you click on that thread, and it presents you with the papers that contain information that can help you understand the transcription factor data that you might have been looking at. Now, some of this data will help you to understand the theory behind what they've done and the types of graphics that you'll be able to see, and they'll show you those in the papers. But there's other data types that you might be interested in as well. So one of the papers that I was looking at the other day is over here. This is functional analysis of transcription factor binding sites. So not only did they give you the stuff from the ENCODE project, but they've taken this further and they've looked at this in assays to see if the data that was being discovered in the ENCODE project is actually relevant in a biological system. And so you can go click this link, leave the Explorer, and read the full paper, and you can see how these folks have evaluated that kind of data and what they've been able to find, and if those signals that you can see in the ENCODE Genome browser actually have a biological impact. I really encourage you to take a look at the ENCODE data. It's there for you. Don't be dissuaded by the drama that's going on right now. There's a lot of really interesting discussions going on, but this data is there for you to check out and see if you can find new and interesting things in genomic regions of interest that will help you further your research. Check that out. Go to our tutorial to learn more about how to use ENCODE, and then travel through the process. Thanks very much for your time.